In Inquiry 8.2, we're going to make a model of a stomatal unit. So our question for this lab is, how do guard cells operate to form a stoma? So think about what we talked about in Inquiry 8.1. What happens to those guard cells that cause the stoma to open? So right here as your hypothesis, make your guess. If the guard cell's blank, then the stoma will open. What do the guard cells need to do to swell up in order for the stoma to open? Okay, our materials for this lab are found on page 131, and our procedures are found on page 135 to 137. Okay, if we take a look at our materials, I'm going to flip to the book. We're going to, in class, be creating a model of a stomata by using a little membrane called a dialysis membrane. The dialysis membrane is a semi-permeable membrane, so that means it allows some things to flow in and out of the membrane, but it only has very tiny openings. Think about it kind of like as a window screen. You know, a window screen allows some things to pass through, things that are small, molecules that can pass through, like air and maybe water, but then things that are too big, like a fly, or something like that, like a bug, could not get through the window screen. So a window screen is semi-permeable. Well, this dialysis membrane we are using is also semi-permeable. So it allows some things to flow through, some things are just too big to flow through. Okay, so we would fill it with water. Now this is not just water, this is actually a sugar solution that has some green food coloring in it. We put green, we dyed it green so that it would be more lifelike with a plant cell in a leaf. And you know, the stomata are found in the leaves of a plant. Okay, so we would put the solution in the membrane. We would use string and tie it off at the top. And it's going to look like this. We're going to place it in a cup of fresh water. Okay, and this is the stomata while it's closed. Okay, if we look at our instructions on our lab, I have a picture there of the closed stomata, or closed stoma, sorry. And then here we have a picture of the open stoma. You can see the opening in the middle, which is the actual stoma itself. These things on the sides represent the guard cells. So we have our guard cells closed. Here we have our guard cells open. Now, what happened to cause this? Well, inside the dialysis membrane that we formed our guard cells with, we have the sugar solution. So it's not just pure water, it's a concentrated sugar solution. It has a lot of sugar in the water. But then in the cup that we placed the stomatal unit in, we have just fresh water, just pure fresh water, no sugar or anything in it. So what happens over time is some of the water from outside of the guard cells gets pulled through that membrane into the dialysis membrane, and it causes the guard cells to swell up. See here, they look real plump, like they have swollen with the water. And so when they swell, it causes a gap between them, which would be the stoma. And in a plant on the leaf, that stoma would allow water vapor to escape. It would allow oxygen or carbon dioxide to escape. It also could allow things to come back in. It could allow carbon dioxide to come in or oxygen to come in and so forth. Now, if we look at this diagram that's on the instructions of our lesson, it again just talks about how this membrane works. This dotted line represents the semi-permeable membrane. Remember, we think about it kind of like a window screen. Little particles can flow through, like water particles, but big particles cannot flow through. So what happens here is the water solution is going to flow into the concentrated sugar solution, and therefore more water is drawn into that concentrated sugar solution, and it causes those guard cells to plump up. This is a process called osmosis. when water moves from areas of low concentration to high concentration, then that's called osmosis. Now, in the opposite way we look at that, the water is high concentration of water. There is a lower concentration of water inside the membrane. So water moves from areas of higher concentration to lower concentration. The sugar, though, is more concentrated inside the membrane. Okay, if we go back and look at our lab report, here in our data, you would draw a picture of what the guard cells in stoma look like before when they're closed, and then draw what they looked like after when they were open. Now, it didn't happen instantly. It took some time for that to happen. So those of you at home won't be live in class to see it happen slowly over time. You're just seeing it instantly, but just know that it took some time for it to go from the before state to the after state. Okay, for your analysis questions, you can use the information that was shared on the assignment to help you answer what happened to the water in your model and what caused this to happen. And how do you think this relates to what happens in the guard cells of an actual stomatal unit in a plant? You know, ours was just a model, but how do you think this relates to what would happen in the actual guard cells of an actual stomatal unit of a plant? Okay, then you have 4 vocabulary words. You can look those up in the glossary. Remember, if it's not listed under stoma, it will have the plural form stomata. Okay, and then for your conclusion, you need to discuss what happens to the guard cells in order for them to open. So go into detail about what you learned about with osmosis in this lesson.